Thanks very much for the kind introduction, and thanks for the opportunity to speak at this series. I've looked at a few of the talks, and that's been some great talks. So hopefully I can contribute something useful. So the title of my talk should really probably be "How much does genome contamination compromise the GTDB taxonomy?" And we'll find out by the end of the talk what we think is the answer to that question. So I'll start with the acknowledgements. I'll start by acknowledging the Genome Taxonomy Database team. They all had played some role in this study. We have the curation team in the form of Marsha and Chris, and then we have the development team, Donovan, Pierre, Adam, who's now no longer with us, and introducing Aaron. So Aaron's actually, this is mainly his work that he's been doing. He's been the primary driver of this. But he's also a developer of GTDB. So the Genome Taxonomy Database is hopefully familiar to many of you. It's a resource which provides a standardized phylogeny-based classification of prokaryotic organisms, bacteria, and archaea. We have been slowing down the releases. We're now at about a yearly release. I think that's probably better because that gives a little bit more time for people to use the current release, and it gives us a bit more time to work out new developments. So the simple goal of the GTDB is to provide a reliable classification of genomes from domain to species. So this is one of the main pages that you will find there. That was the landing page we saw before. So every genome that's included in the database has a page like this, and it gives us information about what type of sequence it is, what we estimate the quality is, a whole bunch of genome characteristics that are either pulled from other resources such as NCBI or LPSN, or features that we calculate ourselves. But first and foremost, it provides this taxonomic string, and it's complete from domain to species, and we also provide the accompanying NCBI taxonomy. And hopefully I don't have to tell this audience in terms of relevance to microbiome, you need to have a stable, reproducible, and accurate classification in order to overlay everything else that you want to do with your data sets. And I would say there's still a reasonably strong need for such a resource like this, and hopefully it will be at one point all standardized and incorporated into longer lasting resources such as NCBI. And the reason why I think it's still needed is that there are still more than 1,000 genomes that are in the GTDB that don't even match at the phylum level, such as this one. So we're saying by phylogenetic inference, this should be a member of the Acidobacteriota, and currently the NCBI, it's classified in a separate phylum. So this is not counting near misses, like if NCBI calls it candidate division W war 3 and we call it war 3. This is actual conflicting information even at the phylum level. So I'm sure many of you would have seen this paper that came out middle of 2021, Orokov et al. And Pierre Bork's group, and it's GUNC, which is, I kept calling it GUNC, but it's pronounced junk. And I guess that's because genome has a soft G, so genome junk. And I guess it's also alluding to the fact that it's there to remove junk. And so in this paper, they look at contamination of genomes, which is a real problem with draft genomes. And they have this very nice figure. I didn't try to make a similar one because they already had a very nice figure within the paper. So this is figure 1, panels A and B. And as most of you will know, if you have an isolate and you're sequencing it, you always have the risk of media contaminants. You always have the risk of laboratory or kit reagent contaminants. And when you go to sequence it and put the genome back together, you can end up, if it's not a complete genome, it's a draft genome in many pieces, you can pick up bits of contamination from those sources. There's been a lot of conversation about MAGs or metagenome assembled genomes, because here you're starting with an inherently mixed community of different organisms. And when you do your assembly and then you bin those contigs out, you do run a risk that you're pulling together pieces from different organisms, so you get these chimeric assemblies. And so this goes into a bit of detail about the different types of contamination, but this is an issue, contamination in general. What really caught our eye in this paper was this statement here concerning GTDB. So after they used GUNC to identify contaminant genomes, they concluded that chimeric genomes likely inflate estimates of total phylogenetic diversity in GTDB, and they estimated that 1,009 species-level clusters in the GTDB, or 3.2% of all species-level clusters in the GTDB, are comprised entirely of contaminated genomes. So that is a terrible statistic. And if you look beyond that, there are other taxa that contain at least one estimated contaminated genome. So of course we were very interested and motivated to check this out, because if that's true, that's a considerable inflation of the taxonomy that's described in the GTDB. So the criteria that we currently use for inclusion, to include them into GTDB, is based on CHECM. And CHECM uses marker genes to estimate completeness and contamination of genomes. So these are marker genes that are conserved at different levels. So here's a picture showing domain-specific markers distributed across the chromosome and clade-specific markers. And then you can see by the presence of those markers, which are highly conserved in the genome, you can estimate how complete the genome is. And if you see duplications of markers that should only be there in one copy, you can then estimate the amount of contamination. And CHECM is widely used by the community, but it's only dealing with a small subset of the genome. GUNC, by comparison, uses all of the protein coding genes in the genome. So it's looking at more of each of the genome. So it's going to, by definition, should be able to pick up contamination that CHECM will miss. And this is another lovely figure from the actual paper, panel C. And I won't go into huge detail about how it works, but it's basically based on sequence conservation to a reference database. So it's looking for similarity of the genes to an existing reference database, and it's looking at all of the genes. And then it uses a couple of metrics so it's a bit more sophisticated than just doing best matches. So it uses a metric like RSS, which is the reference representation score, which is saying, is the closest match to my gene very close or very distant? So it's obviously the closer the match, the more confidence there is. And it also uses this CSS, which stands for clade separation score. And this is looking to see on the individual contigs, so these represent contigs, is there clustering of different classifications. So in other words, that provides additional evidence that there's contamination if you've got a divergent identity for the organism all clustered onto one contig. Now, these scores are then generated per genome. So they're a single score for the whole genome. And the recommendation from the paper is if your CSS or clade separation score is greater than or equal to 0.45 from a scale of 0 to 1, then it's failed. It's putatively contaminated and it should be failed. So that's where we're starting from. So we, or I should say Aaron, applied GUNC to the latest release of GTDB 207, R207, which contains about 317,000 query genomes, which is comprised about 75% of isolates and 25% mags, which surprises some people, but there's an awful lot of isolate sequencing going on out there, so that's why. But this number's slow. This proportion is slowly increasing over time. And then the genomes were assessed relative to two databases provided by GUNC, the in-house one, the ProGenomes one, and then they've also taken an earlier release of GTDB. And as you can imagine, because this is a similarity-based assessment of chimerism, it's going to be very dependent on what you have in your reference database. And we can see this here in the overlap of the estimated chimeras is 10,000 out of a total of 35,000 failed genomes were identified by both reference databases, that is, they're having clade separation scores of greater than 0.45. And you'll also notice from this picture that the number of failed genomes, the proportion of MAGs goes up slightly from, well, from a quarter to basically a third in both cases, which is one of the take-home messages from the original paper that, we should be wary of MAGs. So in total, that's almost 12% of the genomes in R207 are predicted to be contaminated, which in my opinion is a very large number of genomes, almost 36,000. And if we go back to that metric of looking at species that have been defined in R207, whether they're comprised of entirely of failed genomes, we end up with 6%, so double the percent of the initial study, which was based on release 95. 6% of almost 4,000 species clusters, prokaryotic species clusters in R207 consist entirely of contaminants. So we were very alarmed to see this result, as you might imagine. And so if you look at bulk stats, such as the number of contigs between past and failed genomes, you see things that make sense. For instance, failed genomes in general are typically more fragmented. That would make sense because they're lower quality drafts, more chance of contamination from one side or the other. One thing that I didn't fully appreciate from the initial paper, which came out through our analysis, is that of those 36,000 genomes, a large number of those failures are actually concentrated in really well-represented species. So for instance, E. coli, there are over 3,500 genomes from E. coli that fail according to the GUNC analysis. And we see Klebsiella, Salmonella, all the usual pathogenic, very well represented species. And you'll also note that most of these failed genomes are isolates and just this very thin layer of MAGs at the top. But this obviously reflects the constitution of these species, which are mainly populated by isolate genomes as part of surveillance assays and other things like that. So a large number, a third of them just within 5 species, and if you keep going, a large number are occupied within existing species. So that actually is interesting to think about, because if that's correct, when people come to do pangenome analysis, which is a very popular thing to do, particularly for pathogens and well-represented pathogens, it means that if there's contamination, you're probably overestimating the size of your pangenome. You're saying that these are regions of the genome that are unique to a particular strain, and in fact, it could be contamination. So that's something to think about. Not our central question, but something for people to think about. I will point out that in the GUNC paper, there was this sentence which is cautioning against overinterpretation of contamination estimates at the species or strain resolution. And there are reasons because to pull it out, that there would be potential overestimation of contamination at that very fine scale. So getting back to our main question is, how much does it compromise the GTDB taxonomy or inflate the GTDB taxonomy? Does it inflate the GTDB taxonomy? If you take it at face value, then certainly yes. You look at, this is the R207 data. And now we're focusing on the bacteria for the rest of the talk, just for convenience. That's where most of them are. So if you look at this, if all taxa that have failed genomes in them are removed, you would end up removing 3,700 species, 500 genera, 40 families or so, no phyla but 1 class. And so the inflation ranges at species level from 6% down to 0.2% for the class. So that's a significant amount of taxonomic inflation and stuff that basically we don't think exists. So we wanted to ask the question though, are all of these spurious taxa actually spurious? And the way we went about this was by, rather than just removing the whole genome, which I think is the recommendation from GUNC, this is a bad genome, take it out of the running, we went to actually remove the contaminated portions of the genomes and then reclassify the clean genomes. So the first thing we did, or the first thing Aaron did, was that we wanted to come up with a contamination score at the contig level. So GUNC produces a contamination score per gene and then pushes it out and gives you a single score for the whole genome. We wanted to have a score for the for each contig, And this is basically, we use the first two steps of GUNC, identify the matches on the each individual contig, and then classify them against what the most common taxon is taken as the reference taxonomy and how divergent the contig is from that so-called clean taxonomy. So in this case here, you see that the least congruent is this contig, which contains an archaeal gene on it. So that's the least congruent, and the most congruent has a single gene belonging to the dominant reference taxonomy in the firmicutes. So that's how that's done. And then, so again, just looking at the bacterial failed genomes, the 34,846 failed genomes, we order the contigs within each of the draft genomes, the failed genomes, from most to least contaminated. So here's the least contaminated or the most congruent, and these are the most discordant or potentially most contaminated. And then what we do is we keep the clean half, so for further analysis. So our assumption here is that less than half of the genome is contaminated. Now, there could be cases where you have grossly contaminated genomes that are real Frankenstein assemblages of multiple different organisms, but we were making the assumption that the contamination should be less than half of the genome. So that's our first assumption there. Then we take those clean genome halves and we basically put it through our standard GTDB classification workflow. And so that's a two major step process. First of all, we take all of the genomes and using average nucleotide identity greater or equal to 95% and an alignment fraction of greater to equal to 0.5, we cluster them into species by these metrics. And so this reduces the total number of genomes that then need to be analyzed via a phylogenetic tree. And so we then use 120 markers that are conserved amongst bacteria and make a concatenated tree of just the species reps, which are the circular dots here. And then the classification of those is then propagated to all of the other non-species reps within the radius, the ANI radius. So for the bacterial failed genome set, about 30,000 of the genomes are non-species reps and 4,500 or so are species reps. So we analyze those in that order. So first of all, looking at the non-species reps, we simply used ANI in order to classify that clean half. And what we saw was that 99.76, so almost all of these genomes ended up with the same species classification as using the whole genome was. And so an example of this is here. So we're not saying, I want to be very clear here, we're not saying that these genomes aren't chimeric, we're just saying that the contamination is not sufficient to have changed the classification based on the ANI. So here's a good example of quite an egregious contaminated genome. It's Enterococcus lactis, and it's actually got, actually this is the wrong, it's not interphylum, sorry, that's the wrong contamination, but it's contaminated with, it is interphylum. It's contaminated with Bacteroides, which is in a different phylum. But that was insufficient to move this genome out of the valence of the representative sequence. Then we had just a few that were assigned to a different species based on the ANI. And just for breakdown, that's 26 of them are isolate genomes and 19 of them are MAGs. And here's an example. So if you have the full genome, this organism was within the valence of the representative for Anaerococcus vaginalis. Once that was removed to the so-called clean half, then that was now within the valence of a different species within the Anaerococcus genus, Obesiensis. And so, ironically, it's questionable whether this is actually contamination, because both of these, this is basically a very tightly related series of species. And so it could be that this is just movement of this genome between species due to the tight clustering, and may not actually be contaminated. This may actually be not contaminated, but you get this movement. Regardless, the bottom line was that if we had reclassified just the clean half, these would now represent different species of the same genus. So we count these as contributing to changes in the taxonomy. We then had 27 where we had an inconclusive result from ANI, and this is because either the ANI was less than 95% or the alignment fraction was less than 0.5. However, when you put these into a BAC120 tree, they all place within the expected genus. So in fact, if we did make trees from all of these, and all of these guys place within the expected genus as well. So now moving on to the species reps, this is a bit trickier because we can't classify these against the species reps because they are the species reps. So the best we can do is place them into the tree, take out their own species reps, which is the full genome, place the clean half in the tree, and then we can see, do they fall into the expected genus? And again, the great majority of them did do this. So in this case, a bit over 97% ended up in the expected genus. So here's an example. And again, I want to point out that the classification is somewhat independent of the contamination, because here we have actually another very egregious case of contamination where we have a bacterial genome in the actinobacteria that has a couple of contigs that belong to an archaeal organism. But what we see is that this was insufficient to move to make this genome. So the clean half, this is the full genome, the orange part, the whole thing. It's within this genus UBA 8592. And then when we remove the contaminated fraction, most of which is actually still the right genome, so it still falls within the same genus at a slightly different position. And actually, when you think about it, in this case in particular, this is made up of bacterial marker genes. So there was no contribution of marker genes from the archaeal fraction to this inference. So maybe not so surprising. So no change to classification, although clearly a contaminated genome. So 120 of the genomes, or 2.6%, the clean half was classified to a different species. And we could see that based on ANI. So of those, 117 were classified to a different species in the same genus, to a different genus in the same, two to a different genus in the same family, and one was classified to a different family of the same order. And here's an example of that. So this is actually an intergenus contamination between Klebsiella and Citrobacter. So they belong to the same family, but they're different genera. And so it's a fairly large amount of contamination. So you see here, this is the contamination from Citrobacter, and here's the Klebsiella. And so the whole genome in orange was sufficient contamination that it made a new and spurious species here. So in fact, this is Klebsiella planticola and should be here with this reference sequence, but the contamination is sufficient and contributing marker genes and distorting the ANI result enough that this then becomes a spurious species. So this is a bona fide example of taxonomic inflation caused by contamination of the genome. So now revisiting the original putative spurious bacterial taxa. So you probably didn't add up the numbers, but there are 192 genomes with changed or inconclusive classification after cleaning. And if you then now propagate this back to these 192 genomes, they represent the entire species. So the same criteria as these original ones, that they have to be representing that the species is represented by a fully failed genome set. You end up with only 62 spurious species, and that's comprising 42 mags and 21 isolates. So what it's saying is 61 of these spurious species were singletons, so just one representative, and one of them had two. So that represents a 0.1% inflation of the R207 taxonomy. So I just want to go back to this contamination that GUNC picks up. So some genomes should be removed even if they don't affect the taxonomic assignment. So we saw 32 genomes with very clear gross contamination that could be confirmed. 6 of these were intergenus chimeras, 3 into family, 6 into order, 8 into class, 6 into phylum, and 3 into domain chimeras. So we saw that case where we had the actinobacterium with a chunk of archaeal genome in it. And in these instances, actually, mags definitely do dominate these type of chimeras. 28 of the 32 were mags, 2 isolates, and interestingly, 2 sags, because sags are often held up as a way of making sure that you have very clean genome assemblies, but I'd say even SAGs, or can be contaminated with, particularly with, say, reagent contaminants, what have you. And of these 32, 21 of them were species reps, so that's important. Other genomes have been classified against them, and 11 of them were non-species reps. Interestingly, only one of them, which is the one I showed you before, resulted in an actual misclassification. The others actually behaved themselves despite the contamination, and that's because, on average, the contamination is representing, usually not more than 0.3 of the genome, so sufficiently small amount that it doesn't distort the classification. So conclusions then, there are only a few conspicuous contaminated genomes, some real beauties though that were apparent from our tree and ANI based analysis. Even fewer resulted in changes to the GTDB taxonomy. And again, I stress this does not mean that there are not loads of contaminated draft genomes in circulation, but they're too subtle to be reliably detected by analysis, or too few by the ones that are detected. But more importantly for us, they minimally impact the GTDB taxonomy, so we give that a green tick. And we're still sort of debating how can we incorporate this information into our workflow, because actually running GUNC is computationally very costly. And if you look at it in terms of what you get for that additional analysis, it does turn out to be, in terms of classification, a pretty small benefit. But I don't like the idea that there are these landmines sitting amongst the people will do some analysis and they've got this contaminated genome. It really shouldn't be there. So somebody should be doing something about it, whether it's us or not. So finally, all of this will hopefully become a historical footnote. I'm sure everybody in this audience is aware of the effects of long read sequencing, particularly nanopore. Many studies in recent times where we're now getting more or less complete genomes. And so barring bad assemblies, this should preclude the issue of having binning together bits of other genomes or bits, spurious pieces of DNA, as we've seen some examples in this talk. So this is a nice image from the Moss study in 2020, and they show, you get basically with the nanopore thrown in, you get the complete genome back for this Prevotella copri. Okay, that is the end of my talk. I hope you found it interesting, and if you have your questions, now is the time to ask them.